If not, I'll turn it over to Chris to introduce today's speaker.
Great, so it's my pleasure to introduce Kate Laskowski, who just started her lab at UC Davis a few months ago in the Department of Evolution and Ecology. So Kate originally did her undergrad at Maryland, and then a PhD with Allison Bell at University of Illinois, and then spent the last 6 years in Germany, in Berlin, at the Leibniz Institute. Is that right? Yeah. And so Kate does what I sort of think of as the next generation of animal personality sort of studies. Sort of sophisticated mixed model analysis, ask her about your mixed model questions, as well as sort of sophisticated animal tracking over the lifetime of an organism. And then she's worked on everything from pike to stickleback to social spiders to finally, I think her chosen study system are these sort of fascinating clonal fish, the Amazon molly. So take it away.
Great, thank you. Yeah, thanks for the invitation and thanks for being here. It sounds like you guys could probably go to 1 to 2 cool seminars every day of the week. So I'm glad that you're here for mine. Yeah, okay, so I'm a behavioral ecologist and I want to talk to you about behavior. And in order to do that, I want you first to think about this fish. Imagine that you are a fish. And so think about all the things that you have to do if you were this animal, right? You need to find food. But you yourself need to avoid becoming food. You probably need to compete with lots of other species that are in your environment. And at the end of the day, you just want to find that special someone and start a family. And so all of these things that this animal has to do are mediated through its behavior, right? And so for me, I find behavior fascinating because it really sits at the interface between ecology and evolution. As we understand that ecology will shape the behavior that animals exhibit, we assume that we know that these behaviors have fitness consequences and so will affect the evolution. And of course we assume that evolution has shaped these behaviors, and finally we know that the behavior of these animals will shape the ecology of the system. And oftentimes when we think about these behaviors, traditionally in behavioral ecology, we think about them within their certain context, right? What does it take to become a good predator? Or what does it take to be a good mate or a good competitor with other species? But I think it's really important to remember that all of these behaviors are occurring in the exact same animal. Not just the same animal, but the same individual animal. And as soon as you start thinking about that, there's suddenly lots of trade-offs that this animal has to make, right? If you want to be a good forager, you have to do it in such a way that you also don't become prey for some other animal. If you want to be a good mate and be super aggressive and compete with other males, you have to do it in such a way that you don't scare away the female that you're trying to attract. And so there's all these trade-offs now that you have to manage. And there's going to be different ways to solve these trade-offs. And as soon as you start thinking about that, you realize that individuals are different. And they're all going to find slightly different solutions to all these different trade-offs that they have to make. And the cool thing is, we know that individuals vary. I mean, Darwin knew that individuals vary. This is not a bold statement to make. But the interesting thing is that not only do individuals vary, but they do so in a very consistent way. And it's this predictability that we've gotten really kind of excited about and we find really interesting because it suggests that there might be constraints or limits on the behavioral plasticity that an animal can exhibit. And so when I'm talking about individual behavioral variation or consistent individual behavioral variation or animal personalities, temperament, whatever you want to call it, it means the same thing, which is that if you had a group of animals and you measured them multiple times or maybe across different contexts or whatever, you'd find that you'd be able to predict that animal A is consistently, predictably more aggressive, more active, more whatever compared to these other animals. And so when I talk about consistent individual behavioral variation, this is the phenomenon that I'm talking about. And so when I started my PhD, I was really excited about these individual differences in behavior. I thought they were super cool. My committee was less impressed, to say the least. They thought that this was kind of like a one-off thing. They were like, how important, really, are these individual differences? How common are they? And so, of course, my advisor, Allison, is super smart, and she was like, let's do a meta-analysis. And so we called the literature for estimates of repeatability. And repeatability is a great statistic. It just measures the proportion of variation that's due to these among individual differences, so how different animals are. And so the closer you get to 1, the more different these animals are. And luckily for us, lots of people have measured repeatability. They normally do it because they want to measure heritability, but we can use it. And what we found is, one, that a lot of people have measured repeatability across so many different taxa. We found over 100 studies that did this. And by and large, the average repeatability was 0.37. And so this means that about 40% of the behavioral variation is due to these individual differences. That's a pretty big number when we're talking about ecology and evolution, right? I mean, if you were doing a QTL analysis and you found some loci that explains 40% of the variation in whatever trait you care about, that would be a QTL of major effect. And so these individual differences are ubiquitous, they're arguably strong. And one of the reasons that behavioral ecology has gotten pretty excited about these is because we know that individual variation is the raw material, obviously, for natural selection. But it could also be that these individual differences are the product. And so there could be some sort of mechanisms that are generating this behavioral variation within the same species, the same population, the same sex, the same size, the same whatever, are generating these behavioral differences. And so these are the questions that drive me, that I get really excited about. It's kind of the most fundamental question in all of evolutionary biology, right? What drives the generation and maintenance of all of this behavioral variation? And so that is the story that I'm going to tell you today. And there's three kind of chapters to the story that I want to talk about. The first one is I'm going to talk about why I think social interactions are really important. I'm then going to talk about, zoom out a little bit and talk about how these social interactions can then influence an animal over their entire lifetime. And then finally, I'll zoom out a little bit more to think about a general hypothesis about what I think is generating these individual differences, and hopefully maybe offer a novel or complementary perspective. And so this first one, social interactions as adults. So here we have this individual animal. And just as I said, these behaviors don't occur in a vacuum. You have to consider them in the context of other behaviors. This animal also doesn't occur in a vacuum. He occurs in the kind of context of all of these other individuals, right? So essentially, every sexual species that exists has to interact with one other conspecific at least once in their life. So social interactions are ubiquitous, and they're very necessary for fitness in the vast majority of species. And so we think that these social interactions, therefore, might be a key driver to explaining why this behavioral variation is so ubiquitous. And of course, the idea that social interactions are important in influencing individual behavior is not new. We have classic game theory like the hawk-dove game that describes how when you're in a social context, your behavior, the best thing for you to do is going to depend on your own self, but of course what everyone else is doing around you. So we've kind of exploited this classic game theory now to think about not just the instance of behavior in this particular moment, but then why would individuals do the same thing over and over and over again? This predictability, which is really the hallmark of these consistent individual differences in behavior. And I'm just going to kind of summarize the theory here into this main point, which is that if you're looking at a group of animals that have some sort of mutual goal, it's going to pay for them to be different from each other, because then they can reduce competition if they're competing for something. They can also reduce cooperative overlap if they're trying to do something cooperative. And the other really cool thing, the important bit here, is that it's also going to favor them to behave predictably. This is really important. This is like the key ingredient to all of these individual personality, individual behavioral variation models. And this is because as soon as you start behaving predictably, others can reliably either manipulate you or exploit you. And this can help enhance avoidance or coordination if you have some mutual goal that you want to achieve. And so what sorts of mutual goals might animals have? Well, there's a couple. There's two that I'll describe. The first one is a very classic one, this idea of competitive displacement. This is actually an example of a mutual goal. So if a group of animals are foraging in a very patchy environment, it pays for everyone to differentiate and use a slightly different resource, because then you're not super closely competing, which will reduce your individual intake. Another more obvious mutual goal that I think a lot of people can think about is this division of labor. So if you have wolves that are hunting a buffalo or a bison or whatever this is, collectively, it pays for them to all do something slightly different because they're more effective than at taking down this big prey item. The other thing is that in both of these contexts, it pays for them to be predictable because then the next time these animals interact, they don't have to wait to figure out where they should go. They immediately go here to that patch because they know that's where they feed, or this wolf immediately attacks the head or whatever wolves do when they attack bison. And so this was the first theory that I really wanted to test. And it's the idea, it's this first scenario here, which is, does competitive displacement, the opportunity for competitive displacement, actually favor the generation of these individual differences in behavior? And so to do this, we had to create some treatments where we had different foraging scenarios that were different in the opportunity that they had for this competitive displacement. And for this, I used sticklebacks. They're these great little fish. They're really a workhorse of behavioral ecology. People like them for a lot of reasons. I think they're adorable. But for this purpose, they're great because they like to forage together. They're always hungry. They're super motivated for bloodworms. So you can get them to do a lot of cool things. And we created 2 treatments. Well, first let me tell you, I created this crazy feeding machine. It was this big, long tank where I had conveyor belts on either side that you could add in bloodworms at a very precise rate. It took me like 2 years to build this. It was absolutely probably a waste of time, but it worked, and so I'm happy about that. But this first treatment is our control treatment where there's no opportunities for competitive displacement. And what we did is we would have a group of 6 animals in here. They could get bloodworms on one side, and then after 5 minutes, they could only get bloodworms on the other side. So there's no opportunity for anybody to displace anyone here. They all just have to go to the second food pack. This is the control. Of course, the interesting treatment is now this one, where we have the opportunity for competitive displacement. A trial would start the same way, where food is available on one side, and then after 5 minutes, it'd be available on both sides. And so now these animals can displace each other in this competitive environment. And we measured these groups of animals multiple times. I think it was 10 times over the course of 5 days. And the key is not just whether or not they use the 2 food patches, they should, but whether or not the same animals consistently use this new food patch. So that's what we wanted to test. And so the prediction here is that this individual behavioral variation should increase if it helps reduce competition, which would occur here. And so let me walk you through these graphs because they're a little complicated. But so here's the 5 days that we observed them. We observed them 10 times over these 5 days. Here's our 3 different groups. This is the foraging behavior. It's latency to utilize that new food patch. So how long did it take them to get there? And now I'm going to draw some lines on here. Every line is an individual. And you can see is that these individuals here, for example, are learning to utilize this new food resource quite quickly. That's great. That's what we expect. The most important thing, though, is that the repeatability down here is 0. And this is because these animals are all doing the exact same thing. There's no differences among them. But it's good. We've shown that they use this foraging assay in the way we expect. The cool thing now is here, in these graphs, this is where they can now utilize these 2 food resources. And guess what we see? A bunch of lines. But the cool thing is that some of these lines, this individual here, is consistently the one animal that goes to utilize this new food resource. In fact, by the end of the week in this group, you have perfect rank order consistency in what these animals are doing. So they have really become different from each other in their behavior, and they're doing so in a predictable way. And so there is evidence here that individual behavioral variation does increase under these conditions where we think it helps them reduce competition. That's cool. Yeah, and so we think here that individual behavioral variation, like I just said, is favored because it does help them reduce competition. And to me, it's quite interesting if you think about this in a kind of a broader context of just ideal free distribution theory, which is really a classic paradigm in behavioral ecology, is one of the main things about the ideal free distribution is that it assumes that all individuals are equivalent, they're all doing roughly the same thing, and then they all kind of get to the same distribution. But here, this is suggesting that rather the distribution of foragers might just be driven by just a few highly responsive animals that are creating a whole population, group level phenotype. so to me that's quite interesting. The other thing I really like about this is that this competitive displacement is the same thing as character displacement that we see among different populations or among different species. And so it suggests that similar ecological processes that are at work among populations might also be at work within populations. Okay? So now going back to this idea of game theory, I've given you some evidence that when groups do have a mutual goal within a competitive context, you get differences among animals, and they also become predictable in their behavior. The next key prediction from this body of theory here is that then this individual variation, these differences among these animals, should then become stronger the more these animals interact. Right? As you learn who your group mates are, you should figure out your role in this group a lot better, and you should do it more consistently, more reliably. You should build a social role or a social niche, as we call them. And so that's what I wanted to test here. I again use these sticklebacks using the same foraging paradigm that I told you about. And the idea here is that I created these familiar groups. These were sticklebacks that had been living together for 3 weeks. They were getting fed together so they could figure out where everybody likes to feed, find their own role within that group. These sticklebacks went through the same experience, but then I tested them in the same array using animals that they had never met with before. So presumably they wouldn't know what role the other animals like to take. And the prediction is that the individual variation should increase in these animals that had more opportunities for repeated social interactions, right? Guess what we found? Not that at all. There was no evidence that repeatability increased with increasing familiarity in these sticklebacks. And to me, this is me as a PhD student, I'm sure a lot of you have gotten negative results before, and it's super disappointing at the time. And so I was trying to figure out what's going on. I'm thinking about this, and I'm reading all these papers on stickleback social behavior, and I realized that sticklebacks live in really unstable groups. They have lots of fission fusion, they're constantly meeting new buddies, they're constantly interacting. And so the idea that they would have repeated social interactions with the same group of animals is maybe not a very good assumption to make. And so I figured that if these social roles and if these social niches are important, they're going to be most important in a species that lives in a very stable social group. So that is when I swapped out a fish for a brief foray into spiders. And these are social spiders. And so these critters I think are super cool. And they're great for these sorts of questions because they live in the same group their whole life. So they're always interacting with these same animals over and over again, unlike the sticklebacks. The other interesting thing is that they cooperate for many tasks. So they do have mutual goals that they want to achieve. Importantly, there's no, as far as we can tell, there's no morphological or caste specialization in these animals, so they seem to be able to do whatever they want. And importantly, also, all the females reproduce. And so in these spider societies, you don't have this weird kind of eusocial insect where only one animal is reproducing. That creates lots of conflicts of interest. Here, everybody can reproduce, so to me, that's important for these sorts of questions. And so what I wanted to do was ask the same question that I did in the sticklebacks, which is, when these animals have more opportunities for repeated social interactions, do they figure out their social role, and does their individual behavioral variation become stronger? And so I used essentially the exact same methodology that I did for the sticklebacks, but here, as opposed to just having familiar and unfamiliar animals, I had kind of gradations of familiarity. So they either live together for 1 week, 2 weeks, 3 weeks, or 4 weeks. So these animals over here are far more familiar. We also had control animals where they always live with the same animals. And then the mixed ones are the ones where the familiarity was altered. And so I'm going to show you repeatability here of their behavior. And what you can see with the control animals is that they're always repeatable. And this is because they've always been with the same group of animals. This is good. We know that these animals have really strong, consistent individual behavioral variation. So here's the mixed colonies. And so these are the colonies where these animals here have only been living together for 1 week, and these animals here have been living together for 4 weeks. So individual variation does increase with increasing familiarity in these social spiders. how cool is that? I've never had such a clean result in my whole life. I don't know if I ever will again, but I just think this is really fun. The other thing that's cool is people always ask me, okay, so animals are different in their behavior. Does it matter? Yes, it does. And so here we have the change in mass. And this is for the control colonies. We weighed them before and after they went into this treatment. I think it was 5 weeks long in total, so this is a pretty long portion of their life. And luckily, the control colonies, they all gain weight. They should. They need to be, getting bigger so they can have more babies. Contrast that now to the mixed colonies, where actually this mixing experience where they get put in with unfamiliar individuals is costly. They lose weight because of that. Whereas those that have been living together with their same buddies for 4 weeks, they gain weight. So these individual differences and the presence of this individual behavioral variation is important. Yeah, so individual variation does increase under increasing familiarity. And so this is evidence that repeated social interactions do stabilize behavioral variation, at least in these species that have really stable social groups. And we think this is because the individuals are learning their role in the group. This has positive benefits to the individuals, like I showed you with their mass. The other thing I didn't show you is we also did collective behavioral assays, and groups that are more familiar with each other are better able to repel an intruder and capture a prey item. So having these individual differences in behavior has really strong consequences, both for the individual and for the group. And if you think about this in a broader context of sociality, is that this group stability might be acting as a stepping stone for more organized social systems. So as soon as you start interacting with the same animals over and over again, you can get a really strong social role. It's not hard to imagine a world where that then becomes a kind of set role, either through morphological differentiation or something like that. And now if you compare this to the stickleback work that I showed you, both the sticklebacks and the spiders had mutual goals. One was a competitive goal for the sticklebacks, and one was a cooperative goal for the social spiders. However, we ended up at the same result, which is that you get these individual differences, but the social pressure that's creating it differs based on the species ecology. Cool. So there's social interactions as adults. And of course, a lot of you in here are probably thinking, well, social interactions don't just occur in adulthood. They occur all the time. And I'd say you're right. And so this is now what I'm curious about, is that these animals, are developing, and they're of course developing within a social environment. And so social experiences might be really important over a whole animal's development. And how do they accumulate over time? And so social interactions might be especially informative, especially during development, because they offer insight into your environment. They can tell you about your social position, they can tell you about what the level of predation risk is, they can tell you how many resources are available, they can give you a lot of information about what your world's going to be like. The other cool thing is that we know that feedback loops, or experiences that you have early in life, can set individuals down different trajectories. So something that happens to you early might predispose you to go down a certain path. And so this all brings up the prediction, is that early social life experience should predict later adult behavior. And so this is what I wanted to test now, but I had to figure out what early social life experiences do I think are most important, what adult social behaviors do I think are most important. And when I was thinking about this, I watched fish a lot, and I was thinking about dominance. Because when you put critters together, they often fight. And the cool thing about dominance is that we know that there are lots of factors that can determine their rank. So intrinsic things that I like to call, such as body size or your genetic variation or something like that. But also, of course, experiential things, is easier to say. Things like previous experience or social context can affect you. And of course, I really like social behavior, so this has got me excited. The other thing that's great about dominance is that we know that animals practice it, right? They play. And they do a lot of things early in life that we think helps prepare them for what they're going to do later in life. And so looking at dominance behavior seemed to be a really good thing to investigate how early life experience might affect this later dominance behavior. Yeah, so does early life social experience determine later adult dominance rank? And of course, I wanted to focus here on the experience that these animals had early in life. But as you can see here, there are lots of things that can affect your dominance and your rank. So how do you control them all? Well, Chris stole my thunder a little bit by already telling you the coolest thing about this animal, which is that it's clonal. And so this is the Amazon molly. She's a pacillid fish, so she's a live bear. And she's incredible for these types of questions because she reproduces clonally naturally. She produces broods of babies that are genetically identical to each other and to herself. And it's just what she does. It's like her thing. Not only that, but she's also a live-bearing, happy little lab rat of a fish, and so the day these animals are born, I have complete control over their experiences. And so it's just really the ultimate twin study. And I can use her now to ask all sorts of questions about how does early life experience affect later adult behavior in a genetically identical replicate individual. How cool is that? The other thing I'll mention that I really like is she's named the Amazon Molly because she's named after the all-female Greek warrior tribes. She's not from the Amazon. I don't get to do field work in Brazil. She's from Texas, so I get to go to Texas and collect her. But I really, I don't know, it speaks to the feminist in me that I'm studying an animal named after Greek warriors. I think that's cool. Yeah, so here's my question. Does early life social experience determine adult behavior in this Amazon molly? And this is a crazy experimental design, so I'm going to spend some time walking through it. We wanted to see how does your early life experience with winning or losing a fight affect your ability to then win a fight later on. And the good thing about these mollies is that you put two of them together, they'll fight. They like to fight, that's what they do. The other good thing about these mollies is that who wins the fight is almost solely, as far as we can tell, determined by their body size. And so I can now manipulate the body size of these animals to fix what experience an animal will have. Will they win a fight or will they lose a fight? Because that's what we did. We had all of these clones here, and we separated them into 3 treatments that I'll call the winners, the losers, and the variables. It's perhaps obvious why we call them this in a minute. So all of these guys are size matched at the beginning. Every week they'll get paired with a new animal in such a way so that the winners are always bigger. So when they fight, they always win, and they did, they always won. The losers were paired up every week with a new animal that was bigger than them, so they always lost. And then the variables, honestly, we just wanted to just kind of have a gradation of treatments of between always win and always lose, what happens if you have half and half. And so that's what we did here, where 1 week they would be paired up with someone who's bigger than them, so they would lose. The next week they'd be paired up with someone who's smaller than them, so they'd win. And then back and forth for 8 weeks. So everybody had 8 weeks of experience either winning or losing with new partners. After 8 weeks, we separated them. We isolated them into their own little tanks and let them grow up for 6 months. So this is a really long time in the life of this little molly. They become sexually mature at about 4 to 5 months old, and so this is beyond a major life history divide. Then after that, we size matched them and we put one of each of the treatments together and we let them fight and see who wins. Right, and so the expectation is that the winners who've only ever won a fight should come out on top. The variable ones who have half as much winning experience should come out in the middle, and the losers should come out on the bottom. And so we did this for 10 groups that you're going to see here. Here's our dominance rank, so if you're at the top, you're the top ranked, the alpha female. And this is what we saw. It's interesting because our predictions are sort of met, so I'll put a check here. Is that early life winning experience does increase dominance rank if you're the winner. If you're not the, so this is this variable one who's the weirdo, who as opposed to ending up in the middle, she ends up at the very bottom in 8 out of the 10 groups. I'm like, what gives? We don't know. Actually, I take that back. We do know. We have some videos. Unfortunately, for most of the groups, we only observed their rank after I think they'd been together for 5 days, so their ranks were quite well established. But for a few of them, our undergrad was super motivated and took videos of this. And so we watched the videos right after they were put together to see what happens. And it turns out what happens is that when you put these three animals together, they don't all fight in like a big scramble. They pair off and they have these dyadic fights first. And it turns out that the winner, I don't know how she signals it, but somehow she signals that she's the only one who's willing to fight. The variable one is the only one apparently who's willing to challenge her, because these two always fought first. They would fight, and then I guess the winner just has more experience with winning. She almost always wins, so then the variable starts swimming away. And the second the fight is over, this loser female senses her opportunity, swoops in, and starts harassing the shit out of this variable one and pushes her all the way to the bottom. And so I think, we really think that this is what's happening, because the cool thing is that in these two groups, you see the exact opposite pattern. Where in this alpha battle, the variable was able to win, and then it pushed the winner female all the way to the bottom. That's cool. Like, this is this cool study that we did, and then we thought it was a one-off, and now I think we have these really cool results, so I desperately want someone to come work with me to follow this up. So this is something you're excited about, let me know. Yeah, so early social interactions, I argue, are key drivers to your behavior later in life. And this early life social experience may be especially foundational. And kind of interestingly, this unpredictable early life experience might really have detrimental effects later on. So if you think you're a winner and you are a winner, you will be a winner. But if you think you're a winner and you're not a winner, you're really going to be a loser, apparently. And the cool thing, I think, is that the presence of this social group really has a strong effect on the outcome of fights. So most work that's been done on dominance really looks at these pairs. And my guess is that had we just paired these animals up in a round robin, we would have found exactly the results we expected, which is that the winner would win, the variable would come in the middle, the loser would be at the bottom. But as soon as you put them in a more kind of ecologically relevant context where they're all fighting together, you get a very different outcome. And the other thing I want to note is that these winner-loser effects have been documented in lots of different texts, so they're very widespread. But as far as I can tell, the vast majority of these winner-loser effects are known to only last for a few hours or maybe a few days. But here, this effect lasted for over 6 months. That's pretty crazy. I mean, it's a little bit terrifying for anyone who's a parent in the room in terms of this tiny experience your kid has early in life might really set them up on a different behavioral trajectory. Not to scare you, if you are a parent. I'm sure your kids are fine. And so this is now social interactions over the lifetime. I hope I've given you evidence that these social interactions really can accumulate over your life, and early life experience might be really important. And a lot of you are probably thinking, well, social interactions are not the only thing that happens in an animal's life. They also get lots of cues and information from other sources, potentially from their genes, potentially also from their parents in terms of maternal or paternal effects. And so this is now the question that I'm trying to answer, which is, how do animals use now all of these cues, not just social cues, to generate these individual differences in behavior that we see? And so I want to take a moment here to take a step back and kind of think about the general hypothesis that we currently have within behavioral ecology about what's generating this individual behavioral variation. And then I hope I'll offer a bit of a novel perspective at the end. So here we are. This is our goal. We know that individuals differ in behavior. We see it everywhere. This is common, it's ubiquitous. The question is now, where does all this variation come from? Luckily, we have a very classic theory, quantitative genetics, that has been used to explore this on an evolutionary timescale. And it says that variation in genetics and some sort of variation in the environment, these will probably interact, will eventually lead to variation in phenotypes, right? And so if you have variation in G plus variation in E, you're going to get variation in P. And this is what the majority of the theory right now in behavioral ecology stems from, this classic quantitative genetics paradigm. And so when I was thinking about this, I'm like, okay, if this is our most fundamental theory that we have right now to explain these individual differences, how do we test it? How do we falsify it? And I was like, well, what if we remove genetic variation, which I can do because I have the Amazon Molly. what if we remove environmental variation, which I can also do because is born and bred in the lab and I have complete control over her environment. The prediction, according to this, is then that we should remove, by and large, most of the phenotypic variation. We should see the optimal phenotype for this genotype in this environmental condition. So that's exactly what I wanted to test, again, using this Amazon Molly, like I just said. She's a genetic clone. We can raise her in the same environment. And so that's what we did. We had a bunch of pregnant females that after they gave birth, we split their broods into these individual chambers. And so we'd raise a single individual under highly standardized environmental conditions. So as far as we can tell, everything is the same. We raised them like this for 7 weeks, and then we tested them in the standard activity behavioral variation paradigm 4 times over the course of a week, I think. And so this is what we expect, is that the removal of genetic and environmental variation should remove this phenotypic variation. We should really see the optimal phenotype for this genotype in this environment. And I'm talking about it. Yes, that's what we expect. I'm talking about it because that's definitely not what we found. We found, in fact, so here's these graphs that I've been showing. Every line is an individual over the 4 observations. And what you can see is that some individuals were consistently highly active. This is a heat map of their activity. And some individuals weren't. And so even though we remove genetic and environmental variation as much as is possible, we're still getting phenotypic variation. I want to point out, it's the same relative repeatability and absolute level of behavioral variation as you would see in a sexual species in the wild. It's weird. So the removal of genetic and environmental variation does not remove this phenotypic variation. Like, what gives, right? Yeah. Oh yeah, and I just wanted to, I have this up here because I keep forgetting to mention it. We have replicated this now a second time with a completely independent set of fish. So this is a robust finding, it's not a one-off. So the question is, where does all this variation come from? And I mean, I'm being a little bit simplistic here. We're not the first people to show this. There's been several studies now in either genetically very similar or clonal animals that they vary in lots of different traits, now including behavior. And so we're just adding another example of this. And of course, I'm also being simplistic in that there are lots of other sources of variation. I mean, you can decompose genetic variation and environmental variation into lots of different sources, right? You can have epigenetic variation. This is something we can't control at all, at least not yet. There could be microenvironmental variation. I mean, these animals are raised under standardized laboratory conditions, but something might still be different. And of course, then there's things like developmental noise that we also can't control, or more stochastic processes. But to me, the important thing is that we don't just need to keep decomposing these variance components more and more and more and making them fine-grained. These sources of variation are just not really well incorporated into the behavioral ecology literature. And I think part of the problem comes from is that quantitative genetics and this framework that I've been arguably using can really only explain variation that already exists, right? So we know that in these mollies at 7 weeks of age, phenotypic variation is there. Quantitative genetics is great at explaining on an evolutionary time scale what might contribute to that variation. But if we want to talk about a single individual in their lifetime, it can't say anything about it because it's not a mechanistic framework. And what we really need now is we need a complementary framework that can fill in this blank space so we can understand what is driving the generation, the process of this behavioral variation within an animal's life. So we want to open this up. And so that's where I'm at now, is I argue that we're at the point where we can use a complementary framework to have more mechanistic or developmental perspective to behavior. So I really like this figure. It's from Waddington. It says epigenetic landscape. And I like it because he suggests that development can be viewed as a ball rolling down a hill. Now where this ball starts will of course be determined by the animal's genes and genetic makeup. But then where it ends up is going to be a complex interaction between cues that it receives from lots of different sources. The problem right now is that in a lot of behavioral ecology, we don't see any of the hill. we only see the end result, right? We see where this behavior ends up, but we don't know how it got there. And so my goal is to now open up this black box, arguably, to figure out, can we predict, one, can we observe, and then two, can we predict how this ball will end up in any of these 4 channels? But in order to do this, we need 2 things. We need a mechanistic framework that can capture what is going on as it's rolling down the hill. And the other thing we need is we need better access to early life. We need to catch this ball rolling down the hill in the act. And so let me tell you about these 2 key components here in just a few minutes at the end. The first is this mechanistic framework. And this is the mechanistic framework that I'm working with right now, is this idea of Bayesian updating. And so Bayesian updating is a really powerful way to integrate lots of different sources of information. If you're getting lots of different cues, how should you use them? And so the idea is that animals might be using Bayesian or Bayesian-like mechanisms to generate their phenotypes. And so I want you to imagine that you are a little baby fish, and you are born, and you need to decide how should you behave. You know, are you in a safe environment with all your buddies around, or are you in a risky environment, so you need to behave very differently? Luckily for you, your parents, or your genes, probably gave you some prior information about what they think the environment will be like. They say, hey, you are in a risky environment, most likely, please behave appropriately. Okay? But then, it might be that you sample the environment, and you find out that really the most likely state of the environment is that it's quite safe. You don't have any cues from a predator, you've never met a predator, seems like you're probably safe. So you can update your prior information to generate your posterior, you can shift your behavior. And then the great thing about these Bayesian updating mechanisms is that these posteriors will now become the prior in the next time step. If the environment stays the same, you can continue to tailor your behavior to match what the environment really looks like. So these Bayesian updating mechanisms, this framework, I argue, is more dynamic, it's more iterative, it more matches the actual developmental pathways that behavior will take. The other cool thing is that it's predictive as opposed to descriptive. So if you know what an animal's prior information is, and you know what the environment it will experience, because you're an experimenter and you can control this, you should be able to predict exactly where it ends up. This is a major departure from quantitative genetics that can only look at variation that already exists down here and figure out where it came from. So I think this is cool. So that's the mechanistic framework. The other thing we need to really test this framework and see if this is what animals are doing is we need better access to early life. And so we know that phenotypic variation in these mollies exists at 7 weeks of age. I've already showed you that. But it's pretty important to figure out whether or not these differences were there on day 1, because that would suggest that there's differences in prior information, if it's genetic or epigenetic or whatever, versus if that there was no individual variation and then it slowly emerged over time, right? That would suggest that the animals are experiencing their environment in different ways. And so I wanted to be able to disentangle between these 2 hypotheses. And in order to do that, I have to figure out what their behavior is on very early in life. And for that, this is the cool tracking system. It looks decidedly not cool in this picture, but it is very cool. It took a long time to build. And these are custom-built tanks. And these little dots here are Raspberry Pi computers. So these little computers, they're like this big, and they're great because you can program them to do whatever you want. And so we have programmed them to take photos of these tanks every second, all day, every day. And so now, this is what it looks like. We can put fish in these tanks and we can track their behavior all day, every day, from the day the animals are born. I mean, that's awesome. And so we have 2 setups. We have this one setup where we have a divider here, so these animals are being raised in isolation, and you can see one animal is being tracked up here. We have an alternative setup here where we don't have the dividers, and so we have a small group of animals here that are being tracked. And so we're really able now to figure out when do these behavioral differences emerge, and then hopefully later we'll also be able to start manipulating different cues that they get to see how they're responding to them. So I'm going to give you the first results. We completed this big experiment, I guess about a year ago. but it produced 83 million photos. We tracked 96 animals for 16 weeks, taking a photo every second. So it's a ton of data. We're still working on diving through it. Here's the first results, which is the big question we want to answer. The first one is, when does this variation emerge? Here we have the total distance that they moved within their tank in a day. And here's the number of days, so it was 16 weeks in total. This is what it looks like. That's just so cool. And so you can see that some animals, like this pink one, is just consistently more active, right? Yeah. And then other ones are obviously in between. So you have, I think it's just really fascinating here. You have differences in mean behavior. You also clearly have differences in variance in behavior. You have this one animal here that I think is a bit of dust that the parent has been tracking for 16 weeks. So I don't think that's a real fish down there. But the great thing is, look at this. When you have this much data, you can really say that these animals are different. The repeatability is 0.62. That's really high. So these animals are really doing something different over the whole 16 weeks. But of course, the thing we really care about is day one. Like, when are these differences there? So I just want to zoom in right now on this first day post-birth. So these animals were born, and on the very first day, look at that. They're different. They're totally different. I mean, this is pretty impressive. This animal is constantly swimming around the tank, and this animal hardly at all. So when does this variation emerge? The first thing I can say is apparently very early on, right? From the day these animals are born, they're doing different things. And so the next steps now, of course, are to figure out, well, this variation is there on day one. What's happening before they're born in terms of, I think maybe maternal effects could be going on. So can the moms inform their babies about different things? How does her experience affect their behavior? These are all the questions that I'm getting excited about right now. And of course, the next big step will also be to test these key predictions from this Bayesian updating theory to see if that's how animals are using information in their environment. Yep, and so just to sum this up, the one thing you take away from this talk is that individual variation should be the null expectation. So I don't think we can't get rid of animal personalities even when we try. So the fact that a group of animals exhibit personality should not be surprising. Rather, I think the interesting questions are what shapes those personalities, what causes them, what consequences they have. And probably these individuals are always different from each other is because they're integrating all of these different cues coming from their genes, from their parents, from their environment. And I hope I've shown you that social interactions might be especially informative and especially critical. And that I think stochastic processes are likely underestimated. Right now within behavioral ecology, we really think about things in a very deterministic way. You have these genes and then you have this experience, you will behave like this. But clearly there's lots of noise in this behavioral variation, and I don't think we're appreciating unpredictability and uncertainty and probability quite as much as maybe we should be. And finally, I really think that this Bayesian framework offers a cool, complementary, predictive perspective on behavior to help get at one of Tinbergen's questions, the mechanisms of behavior. How is it developing over an animal's lifetime? Which will hopefully give us insight into why individual variation emerges and in response to what cues. Yep, so here we are. Just to sum this up, this is where I am now, is I'm really curious about how animals are using and integrating information from all of these different sources to help us understand how behavior is sitting at this interface of ecology and evolution. So with that, I want to say thank you to all my collaborators. So are you thinking, so I can see this going two ways. One, I want to do a breeding experiment with clonal animals and see if you can actually select on behavior even in a clonal animal. Yeah. But are you talking about that, or are you talking about just putting them in the group and then seeing how they respond within their lifetime?
The individual that moves the most distance in each replicate, and now put them together as a group, a new group.
I have no idea. My guess is it probably depends a lot on the costs and benefits of the behavior, if it pays for them to be active or less active.
I would think you'd learn
that's a good idea.
from that.
Yeah, that's a cool idea. I will try that. It would be easy to do. Yeah.
So in that last experiment, they were all genetically identical?
Yep.
Okay. How long is gestation?
About 30 days from the time that they get sperm. Yeah.
So a lot could be going on during gestation.
Exactly. Yeah. So these guys aren't placental, so there's not active provisioning from the mom, but the mom does hold the eggs until they're born. So we have no idea what's going on in utero. There could be lots of cool things. Yeah.
So I love your framing of competitive displacement and cooperative overlap as two potential reasons for reducing this as reasons for behavioral variation, but they have the same prediction. So how in a system, like social spiders, I study them too, and like we make the assumption that it's all about cooperation, but there's probably competition going on in there too. Are there ways of disentangling those that you thought about, or is that like?
I have not thought about that. I would like to think about that.
Because that seems like a great system in which to actually think about it. Because they are still competing for the food.
Yeah, I mean, they want to have their nests survive, so they have somewhere to live, but then of course they still want to get the most food if they can. Yeah, I don't know, let's sit down and think about that. There's probably some ways we can disentangle that, though. That would be cool. Yeah. Yeah.
Going back to your experiments with the sticklebacks in which they experienced different amounts of winning and losing,
Yeah.
do you know anything about how that life experience translates into reproductive success?
No. So it is interesting. So part of the reason we isolated them for so long was that at the end of the 8 weeks, there was dramatic size differences. And so we didn't want to put these animals together that had different sizes. And so that's why we isolated them, to let them grow up. And then when we put them together, they were all the same size again. But I wonder if there's compensatory growth going on. I would love to repeat that, and see if there's consequences in terms of how many babies they have, how big they are, when they become mature. We just don't know. Yeah, that'd be cool.
So in the tracking experiment, when you're looking at total distance moved, so it's just one camera, so I feel like it's a horizontal plane projection.
Yeah.
So rather than looking, because you can't account for the Z in the Y direction.
Yeah.
Have you thought about using like total area covered rather than total distance?
I guess we haven't thought about that. It'll probably be tightly correlated. We do keep the water level fairly low to limit up and down movement, and also that kind of mimics where they grow up. They hang out in shallow water because they can't get eaten. Yeah, I don't know, we could do something like that. We do find that most of our activity measurements are all tightly correlated, so I don't know if it would be a totally different story, but that would be something maybe we can get more fine scale nuance in the data. Yeah. Yeah.
What's the variation in the number of offspring birthed, and does that correlate with their repeatability or
Yeah. Yeah, so there is pretty, I would say, impressive variation among broods. So I've seen broods as small as, say, 4 or 5 babies, and then some broods are 40 to 50 babies. We have included in all of this stuff, I do include brood size as a covariate. It doesn't seem to pop up as something that's important. And we also had a student who was trying to measure body size at birth among these different broods to see if there's differences there, and we couldn't find it, but I think that is because our camera wasn't high res enough, honestly. So that would be something. I mean, you can imagine if you're from a brood of 4, you're probably bigger than from a brood of 50. So yeah, that would be something to look into. Yeah.
So in other highly social species, prolonged periods of isolation have effects on their behavior.
Yeah.
And it seems like a big part of your experimental designs include long, if not like indefinite, like whole lifetime periods of social isolation. Have you investigated any effects of that in your work?
Yes, so that is always a concern, and honestly that's part of the reason I like fish. At least these fish don't seem to be affected too much by it. In that experiment I showed you, the first one where they showed behavioral variation at 7 weeks of age, we also raised them in those small groups, and as far as we could tell there was no difference in the behavioral output. So we're using that as evidence that they're not super strongly affected by it. We also raise them, they can smell the water from everyone. We always mix the water in the sump, so they're getting cues that other animals are around, and we're hoping that makes them feel safer. As far as we can tell, they don't seem to be negatively affected by it, but that's not to say that something couldn't be going on, yeah. Yeah.
Yeah, I'm curious, do you think population success is governed by behavioral variation?
I'm sorry, can you repeat that? I just couldn't hear it.
The success of a population through time,
Yeah.
we were able to follow that 500 or 3 populations through time. Having that behavioral variation, which is your subject, behavioral variation within a group.
Do I think it would make a population more successful?
Yes.
In evolutionary time?
Yes.
Yeah, sure, why not?
Does it?
Yeah.
How do you resist then thinking about group selection? That is, one population does better, a group, for certain characteristics versus others. Therefore, we should look at the whole set of populations and their behaviors.
Well, so I think these mollies are interesting because they do have lineages, right? And so there is evidence of clonal sorting, which is, new lineages are popping up through mutation, or occasionally they get introgression from the paternal DNA. And so you could imagine a world where if you're a lineage that gets a deleterious mutation, you go extinct. So I don't know if that's necessarily group selection, and that there's no altruism, it's just what's happening at the lineage level, is that some lineages, some genomes are more fit than others and are more likely to persist. I'm thinking a lot about, so we do have all these different lineages, and because they are a clonal animal, they have no way to create variation genetically, and so I wonder if they're doing something epigenetically in terms of they might just have more sensitive epigenetic mechanisms that alter the expression of their genes. It's kind of a wild idea, but I'm super excited to look at it. I don't know if that's getting at what you're asking, but I do think there's the opportunity for clonal sorting and selection at the lineage level, certainly.
Maybe we can have one more
I'm
question.
sure your interest is not entirely devoted to this very informative, clonal selection model that you showed. I'm sure your thoughts must still, as informative as that is, vary beyond that and think, we go over most organisms don't have that.
Yeah.
Let's take one more question here from.
Are you certain that there's no recombination?
As certain as we can be, in that we do genetic testing of our lineages every, 3 to 4 generations, and for the markers that we scan, we see no evidence. Other people have looked at this as well and see no evidence. So I don't think there's recombination that's going on in any significant amount, But, so can I expand? Yeah.
Sure.
The clonal molly is super interesting. So it's clonal, but it is F1 hybrid evolutionarily. So it's a hybridization between the Atlantic and the sailfin, so it's perfectly heterozygous. And so we actually think that contributes a lot to its longevity. It's because it has this hybrid vigor. There's not any crazy recessive things that are exposed. Everything's hidden. So we think that's contributing to it.
Wonderful. Well, let's thank Kate one more time.